Good morning. Hello. Welcome to our channel. Today we are going to review a flow cytometry. It's a bone marrow flow cytometry from a patient, 79 years old, male, with a stage 4 DLBCL, a diffuse large B-cell lymphoma, which had a previous bone marrow involvement a few months ago, and is under treatment in a 4th cycle with R-mini CHOP. There's also mild neutropenia, mild anemia, and mild thrombocytopenia. So this is the way we review our flow cytometries. This case is this timeline against the CD45 just to verify the quality of the acquisitions. This is the main first plot that we use to try to identify populations of cells by CD45 against site scatter. This is used to exclude the duplicates, and this is the one that the plot that we use to exclude the debris. Here we have our characterization of the populations. Here is the lymphocytes, which are high CD45, a low site scatter. In between, there is the gate for the monocytes, and the granulocytes have a high site scatter. We use a different strategy than most of laboratories to separate the blast gate or CD45 dim. Under this analysis, we can see that there are a few events in the blast gate, just 1% or something. Granulocytes are like 53%, lymphocytes are 27%, and the monocytes are 74%. Here we have some other plots that we use to characterize cells. Cells are separated by colors. Green are the granulocytes. This deep purple are the monocytes. The lymphocytes are blue, but as the subtypes of lineages are also colored, you will see that there are some lilac and some orange right there. And the red one is the plastic gate or CD45 dim gate. The first tube that we use to follow this case includes separation between kappa and lambda. This is applied to the lymphocytic B cells that are characterized by CD19, and in this case, we can see that clearly this population, there's some certain population here that is kappa restricted, which is abnormal. And from the very beginning, we are seeing that these bone marrow is still involved by this lymphoma. The population of interest, meaning the lymphocyte, the atypical lymphocytes, are CD10, CD5 negative. This way we can separate the T cells that are CD5 and CD3 at the same time. CD38 is not showing any remarkable, except that you can see certain positivity that is then positive as expected in mostly in the monocytes, but there's no other surprise there. Here we can separate the B cells that are CD19 positive, CD3 negative, and DNL is for the double negative lymphocytes, that in this case are a bunch of them right there. CD34 is negative, showing us that there's no primitive cells or precursor cells. CD20 is negative, in the population of interest, and that's because this patient has that expression knocked out because of the treatment. Remember that he's receiving rituximab. Here's CD3 again to verify that we have separating in an appropriate way the T cells. This is CD45. And this is a this is the statistics. We can see that we have 27% of lymphocytes. Just 5% of them are B cells, but the ratio kappa lambda is high, is almost 925 to 1, meaning that this has a residual involvement of the bone marrow by the mentioned before DLBCL. Here we have a second tube, the same strategy. We can say also that these cells are CD43 positive and CD23 negative, which is also an abnormality. This is already seen. This is as well as the one seen before. Is CD2, this population is CD200 negative and 81 positive. There's no other feature that is catching our attention. So in summary, this is a DLBCL stage 4 case in an elder patient whose previous immunophenotype by flow cytometry in bone marrow showed certain involvement, is under treatment in several months after still involved. So this report is gonna be signed out as a positive involvement by lymphoma cells that are under follow-up in about 2% of the events of the analyzed in this study.